We're trying to build the future here, and we believe that biology is going to play a critical role in that. And as Steve and Jeff said earlier, we're a very small agency, and BT only has 9 program managers. And so what we want to hear are your ideas for what we should be building. What things do you think are possible? So if you have an idea that you want to build at DARPA, come be a program manager with us. Take 3 years of your life to come and change the world. If you want to get to work and you want to be all in on some of the ideas that you're hearing today, come and work with us, whether you're in a company or whether you're in academia. And if you have ideas for how to commercialize technologies that we're building, reach out to any of us. We're very easy to find, and we want to work with all of you in making sure these technologies move from the lab to broad scale globally. Thank you for coming. Here we go. Wrong presentation. But I'm happy to give Jack's presentation. All right, here we go. So when most of us think about advanced technology, and especially when you think about DARPA and the awesome things that we are building, you're going to think about hypersonic vehicles that can get from one end of the world to the other in less than an hour. You're going to think about autonomous robotics in the DARPA Robotics Challenge. You'll think about virtual reality headsets that can take you to a completely different universe. And you'll think about the Internet of Things, which is connecting everyone and everything together. But there's another technology that's out there. Biology can do things that no other man-made technology or chemistry can do. It can start from a single seed, and with the genetic code contained in that seed, grow into an amazing complex organism. It can replicate itself. It could scale from 1 to millions to billions in hours. It can adapt to its environment. It can evolve increasingly complex functions. It enables us to learn. It enables consciousness. These things are magical. Imagine if we could harness them and we can use them as a technology. But the challenge is that to date we've lacked the tools and the know-how to do so. And as we're moving out of the 20th century and building the 21st century, many of the technologies that we relied upon to take us to where we are today aren't going to be able to take us to where we need to be. We're facing global challenges to which we have no technological solutions. We're coming to the end of antibiotics, where even the most simple infections can destroy us. We only have about 24 vaccines today. 24. Do you know how many infectious diseases that we have and how many are continually popping up around the globe? We saw it with Ebola. We saw it with H1N1. We're seeing it with MERS today. We don't have the protection that we need for even the things that we know about. And even in a completely different realm, one that I'm from, material science. Materials are the thing that changes our world. If you think about it, we had the Stone Age, we had the Bronze Age, we had the Iron Age, and we're living in the Silicon Age right now. But we haven't had a materials revolution since the 1950s and 60s, and that's holding back the pace of technological advancement. We need a new technology vector. We need capabilities that we don't have today, and this is why DARPA formed the Biological Technologies Office. We saw an opportunity to harness all the advancements that we've seen in engineering, in the physical sciences, in computer science, in data analytics, and combine it with the very rapid pace of advancement we're seeing in biology today, that we're seeing in genomics, that we're seeing in DNA editing through CRISPR-Cas systems, that we're seeing in the neuroscience that Jeff and Philip both talked about. And we see an opportunity to bring these two fields together, to bring all the advancements that we have in the engineering world with the amazing capabilities of biology and build a technological capability that goes way beyond anything we've ever seen today. Biology is technology, and that's what we're here to build. When I came to DARPA 4 years ago, I came as one of those program managers. I was then punished and put into a management position. But I came to DARPA because I was frustrated that we weren't having a materials revolution. I came from the world of nanotechnology, and our goal there was to build matter up from the nanoscale, controllably and reproducibly, to create materials with properties that we had never seen before, but which we knew must exist. And we were failing. We didn't have the tools to do what we needed to do. But biology inherently does this. Biology inherently builds materials up from the nanoscale, from the nanometer to the millimeter to the meter. And it builds materials that way surpass anything that we have in our synthetic world. If you take, for example, our skin, this is one of the most amazing materials that I know of. It senses our environment, it grows with us from birth, it maintains a microbial community, it regenerates itself every 2 weeks, it self-repairs. Imagine if we could build materials that had those properties. Now, the way that we've been going about this so far is we've been building increasingly complex machines, increasingly sterile environments, and at increasing cost. But biology offers us a much more sophisticated manufacturing platform. Take this yeast cell. It's programmable through the genetic code, and it's reprogrammable. As I said earlier, it can scale, it self-replicates itself, it makes billions of tiny little factories cranking out products, ones of which we love, such as beer. And it works in the dirtiest of environments. It works at standard temperature and pressure, and it self-repairs itself. This is the manufacturing platform that I wanted to figure out how to use to build new materials. And so we at DARPA started the DARPA Living Foundries program, and it had a very simple goal. It said, let's leverage biology, not just as a technology, but as a manufacturing platform. And let's go beyond that very small sliver of materials that we're going after, that sliver of biochemicals, biofuels, and a few pharmaceuticals, and let's capture the full complexity of what biology can accomplish. Let's figure out how to make new types of polymers. We've been talking about making a better Kevlar for the last 50 years. Well, let's go and do it. Let's build new catalysts that can do new chemical transformations for very low cost. And let's go to the whole other end of the spectrum and build living materials. Materials like our skin that could be programmable, that can self-repair. Imagine if we could build this. The challenge, though, is that when you look at how we manipulate biology today, it hasn't changed much in the past 100 years. We're still using highly trained human beings to pipette colorless liquids around. We're moving at the pace of humans. And when you move at the pace of humans, your innovations only come in generations. It's no wonder we haven't made much progress here. I don't know of any other technological field that looks like it did 100 years ago, let alone 20 or 30 years ago. And it's not that biologists don't want to change the way they're doing things. It's that until roughly just, I would say, a decade ago, biology was largely an exploratory science. We wanted to discover the fundamental rules of nature. And what I'm talking about here is not exploration or discovering. I'm talking about forward engineering. I'm talking about how do we engineer biological systems to make useful products. And to do that, you need to reorient the entire framework that we've built around biology. We don't need tools for research, we need tools for building. And this was the key insight. I wasn't going to run a biology program, I was going to run an engineering program that allowed us to build an engineering framework for biology that was going to allow me to write down a design that I wanted to implement and allow me to build that design with high accuracy and execute it and run it and learn from it and build something. I wanted a design, build, test cycle for biology. And this is the most important thing I think we can focus on right now for biological technologies right now. Because what happens is that once you begin to be able to bring all of miniaturization and automation and data science to biology, you can start to go around this loop very quickly. And not only just go around very quickly, but you can do tens of thousands of experiments. And when you do tens of thousands of experiments at very high accuracy and very high reproducibility, you can start to apply data analytics to that problem and learn from each time you go around that cycle and go around with much more insight the next time and reach your end goal. And so what I want to do is take you through 4 different technologies that we have been building with our partners here at DARPA to enable us to forward engineer biology. The first of these is that I want to be able to construct my genetic design at low cost, at high volume, and make it accurate. It might blow your mind to understand that less than a few years ago, this was not possible. I could only make the simplest of programs, hello world. And in fact, that would often be garbled, hello world. That's what your DNA code would actually say. And I probably wouldn't even know it until the end. I wanted to change this paradigm. So working with Twist Biosciences, we decided to go from using the 96-well, large volume, low throughput plate and leverage something that we're all familiar with, silicon. There's been billions of dollars put into silicon micromachining, and it offers a way for us to very easily get to 100x increase in throughput, 100x increase at least less in cost, and to do process control so that all of my designs are accurate, and I can, for the first time, write my genetic code at low cost, have it available quickly, and have it accurate. This will change the game completely. The second major technology that I wanted to invest in was to address the issue that we have roughly 30 million to maybe even 30 billion, who knows, that it's not quite understood, organisms on this earth. We use two right now, essentially, for engineering biology, yeast and E. coli. We can do better than this. I want to be able to use any organism that has the properties that I want, whether it's temperature tolerance or pH tolerance, and I want to be able to look at that organism and quickly map it so I know where to engineer it. Now, the challenge with is that today, if you look at our genome annotation software, it's not built for looking at the organism from a systems point of view. It's built for looking for the esoteric and interesting thing that I can publish an academic paper on. So what I'm showing right here is that what we've done is we've taken the millions to billions of base pairs and we've laid them out linearly, and then we're zooming in at about 500 base pairs to look at this little chunk of DNA. There's no way we're going to be able to engineer anything complex off of software like this. But working with Amyris and their computer scientists and bioinformaticians, what we were able to do is actually build what is essentially a Google Maps of genomes. So I can, for the first time, look at an organism from a systems point of view. I can look for the spots in that organism that I understand, the functions that I know, and where I might be able to engineer it. And I can look at it from a very high-level point of view. So what you're going to see here is that we're going to look at all the meiosis data across the organism and where it's located and where I might be able to actually insert new function. Now, with this capability, what I can do is I can take raw genome information and within a day understand how to engineer that organism. So now I can open up the full palette of organisms that are available to us. Game-changing again. The third challenge, and I think what you're all familiar with, but you probably see it as a benefit, is the torrent of genomic data that we're now collecting due to the very rapid decrease in the cost of DNA sequencing. And this is awesome, except for the fact that these databases are just that, data. It's not knowledge, it's not information that we can actually use. Very little of that genetic information is actionable. Only that which occurs in a scientific publication, we might be able to understand how to use it. And so if I'm a genome engineer, these databases actually are useless to me, even though they contain all of this information. So working with two scientists out of Berkeley, we decided to build a computational tool that enabled us to scan through all of these genetic databases, as well as all the PubMed papers, and start to actually annotate this information and allow me to build with it. So for example, what we've been able to do is map the full biochemical space that's accessible using biology. So starting from the center and building outwards, I can now figure out how to reach any biochemical that I might be interested in and all the different genetic paths that I can take to get there and begin to compare one route over another. So for the first time, I can actually do rational design of a genetic pathway. And what's even more interesting is I can sort through these huge databases and begin to understand, for example, here, I'm looking at the World Health Organization's list of essential medicines. And I can understand which of these I might want to target because I can make them at lower cost and make them more accessible through a biosynthetic route rather than a traditional chemical route. Now, the last technology that I want to talk to you about is one that I think could be the most transformative, and that is understanding how do we change the way that we actually do biology. Essentially, automating and making programmable the lab bench. What we're doing here is we're leveraging the trillion-dollar investments that we've seen in the semiconductor industry and the display industry, as well as microfluidics, to basically make these little programmable lab benches that allow us to do tens of thousands to millions of experiments at once, at very low cost, at very high throughput. What's powerful about this platform is not just the throughput capability, the fact that I can, for the first time, do tens of thousands of experiments, but the fact that it's programmable. So whenever I code up an experiment, I can share it with you. You don't have to be the best pipetter, the most accurate person in the lab anymore. We can now share across time and scale different types of genetic programs, and you don't need to be an expert, and we can make things that are reproducible, and we can build off of each other's experiments. This is what led to revolutions in the computing industry, a widely distributable platform, and one in which you could build off of other people's designs with a very low bar to entry. Now, what is the impact of these tools that we are building? So just 3 years ago, prior to the development of these tools, if I wanted to build a new bioproduct, it would have taken me tens to hundreds of millions of dollars in nearly a decade for each product. DuPont 1,3-propanediol, 3 carbons, not complex, $250 million in nearly a dozen years. We can't afford that pace of innovation if we want to have a revolution here. But after our program, after building just this small subset of tools and integrating them together, we've been able to bring down that time and cost by over an order of magnitude. Let me say that again, in less than 3 years, over an order of magnitude of progress. That's amazing and astounding, and imagine how much further we can bring that down. Now, there's still work to do. As I said, 3 carbons, that's not very complex. We need to capture that full complexity of biochemical space. If we really want to make living materials, we have to learn how to make complex programs. And that's what we're working on today. So where is this all headed? Well, before the 1920s, we actually did rely on biology for our materials. It was things that we could harvest from the natural world and we could hone according to our needs. But then in the 1920s, we saw a revolution. We saw petrochemicals come on the stage. And for the first time, we could get new materials properties that nature didn't give us, and we could design materials according to our needs. We're now at a new inflection point where we can think about taking all that complexity and dynamism that biology offers us and combining it with the ability to design according to our needs and open up this whole new materials realm. This is what we're trying to do in Living Foundries. But these platforms, because the language of biology is universal, everything runs off of the genetic code, these platforms can do far more than do a materials revolution. They can transform and solve problems that we haven't been able to solve. As you'll hear later from Dan Wattendorf, we now have the ability to think about providing people immediate immunity against any infectious disease, whether we've seen it before or not. We have the ability to go after vector-borne diseases, malaria or dengue. There are over 3 billion people in this world who are at risk of a vector-borne disease. Mosquitoes kill over 1 million people per year. These tools, for the first time, allow us to go after that problem and for the first time offer us a solution to truly eradicating malaria and dengue and chikungunya and Zika virus, all things that are spread by invasive mosquitoes. We can think about going after resource constraints, as we've seen with the drought in California, or as Martine talked about, when we scale to 10 billion people on this globe, we're going to need technologies that enable us to truly attack these problems at a global scale. After man-made or a natural disaster, maybe we can actually, for the first time, think about truly recovering, completely recovering at a regional scale, the environment. And even bigger than that, for the first time, we have a technological toolkit that allows us to transform hostile environments, not just here on Earth, but could enable us to go into space, not just to visit, but to stay. And that would be a revolution. Biology is technology. We need your help to build this future. We'd love for you to join us. Thank you.